Okay, so today we're very happy to have Dr. Anoop Arunagiri with us, coming to us from University of Michigan. And his talk is Contributors to Proinsulin Misfolding in Pancreatic Beta Cells. He, just a little bit about him, his bio. He is a research investigator at the University of Michigan, specializing in biochemistry, biophysics, and cell biology with an emphasis in protein folding and folding aggregation. His postdoc experience was a research investigator 2021 to present, Division of Metabolism, Endocrinology, and Diabetes at the University of Michigan in Dr. Peter Arvan's lab, and a postdoctoral research fellow, Division of Metabolism, Endocrinology, and Diabetes, University of Michigan. His mentor was again Peter Arvan. So he's got a couple of papers, and his current research is really focused on understanding proinsulin misfolding in pancreatic beta cells and its relevance to diabetes, both type 1 and type 2. And I will be dropping a couple of his papers in the chat. You are welcome to drop questions in the chat, and we will answer them in context of the talk as we go along, but also we'll have some time at the very end. So welcome, and thank you very much for joining us, Anoop.
Thank you. Can you hear me okay?
Yes, sorry, I'm just like also letting a bunch of people in.
Sure, yeah. Hi, everyone. First of all, thanks to Monica and Sugar Science to inviting me to give this talk. And thanks for that introduction. I'm going to today discuss the various contributors of proinsulin misfolding in beta cells. So we here in an R1 lab at Michigan, we do diabetes research. So I'm going to start with diabetes. As you can see from this global map, diabetes is a global burden. Many of the nations are colored here, and the more darker the color is, the more percentage population of people affected by this metabolic disorder.
And just to qualify, this is type 1 and type 2, correct?
That's right. Yep, that's right. So this has been a challenge for researchers and physicians alike. In order to find a cure or treatment for this, we need to understand this disease better. Okay, in the United States alone, 11% of the population have diabetes. 1 in 5 adults have diabetes, and they don't know they have the disease. The major known causes for diabetes are diet, lifestyle, genetics, autoimmunity. These have well been studied, and there are many ongoing nice work, and all of these cause diabetes. One area which is not very much appreciated is looking at diabetes from a protein folding standpoint. Our lab studies that, and we more specifically look at proinsulin folding problems in diabetes. So before getting into what's proinsulin and what are the folding problems, I would like to introduce you to insulin. Insulin is a small peptide hormone that is made in pancreatic beta cells. So beta cells reside in these small islands or islets of Langerhans, which I'm going to simply call islets from here on. These islands are scattered throughout the pancreatic tissue. And within this, we have beta cells, alpha cells, and delta cells, each of them producing their respective hormones. Today, we'll be focusing on the beta cells. That is an electron microscopy picture of the pancreatic section. And we see that there are like thousands of those little granules. Those white circles are the secretory granules that are stored in the beta cells. And there is about 1 billion insulin molecules per beta cell. Each beta cell makes about 6,000 new insulin molecules per second, which means it does really a lot of hard work making this insulin. So what does it do with that insulin? It secretes them out when the blood glucose goes up. When we have a meal, our blood glucose rises. So beta cell releases the insulin and blood glucose goes down. So the function of beta cell is basically making insulin and secreting insulin. It keeps doing that. In a diabetic scenario, you can imagine the blood glucose goes beyond control and beta cells eventually fail after doing all of its hard work. So what's protecting us from hyperglycemia and diabetes? It is the insulin. Where does all this insulin come from in the beta cells in the first place? Insulin comes from proinsulin. Proinsulin is the only known precursor protein, prohormone, that makes insulin. Proinsulin is synthesized in the beta cells, and right after synthesis, it enters into this organelle called endoplasmic reticulum, or ER. I'll be calling that ER. So it enters the ER where it starts folding. It folds to this kind of structure. I have made it very simple for easy understanding on the left. So this is how proinsulin, a folded proinsulin looks like, a B chain, A chain that has disulfide bonds, and there is a C peptide or a connecting peptide that connects the that connects the B and A chain, the dashed line you see there. Those three disulfide bonds you see there on proinsulin that's connecting B and A chain, those are important for its structure. We'll talk more about those disulfide bonds later. Only if proinsulin folds to this structure, which we call as native structure, it can go forward from the ER to the Golgi via these vesicles, the COP2 coated vesicles. It goes to Golgi, and from Golgi it goes forward, and they enter the they enter the secretory granules. During this process, when they go from Golgi to the secretory granules, proinsulin is converted to insulin. So this is the difference between proinsulin and insulin. C-peptide is in proinsulin. We lose C-peptide, what you have is insulin. So that is the biologically active form of the hormone. Insulin shares proinsulin with structure on these aspects. B chain, A chain, and the disulfide bonds, they are still there. Okay, so what we understand from this, we understand that for insulin making beta cells, proinsulin folding is a critical step. Not all the times proinsulin folds well to its native state. There are some situations that lead to improper folding or misfolding. That is what we are going to talk about today. What leads to this misfolding condition? Okay, so today I'm first going to discuss two major contributors to proinsulin misfolding. One is the insulin gene mutations leading to misfolding. The second one is unfavorable folding environment. So I'll introduce this little cartoon. This is not as simple as the way I'm showing it here, but let me explain. On the left, we have a condition where proinsulin folds well. If proinsulin folds well, it can traffic forward from ER to Golgi and then make insulin. Some of them also great. On the right, this is a condition of misfolding. Misfolding either due to a mutation or unfavorable folding environment. In that case, what happens? Misfolded proinsulin stays back in the ER. It cannot move forward. Trafficking is hampered. That's why that symbol, you see that cross symbol. So insulin making is affected, and that can be related to diabetes. So now let's go over each of these factors one at a time. Insulin gene mutations. So our lab has been studying this rare form of diabetes called mutant insulin gene induced diabetes of youth, or MIDI. In this, as the name suggests, the patients affected by this form of diabetes are very young, 3 months, 6 months old, as early as that. So here, the gene mutation is going to cause a structural defect in the protein product proinsulin. Therefore, proinsulin misfolds. Before getting into how the mutation leads to misfolding, I want to just show you the overall distribution of these various mutants on the proinsulin structure. I'm not naming those mutations. I'm just giving a color code. Either they are non-cysteine mutations or cysteine mutations. So cysteine mutations are obviously going to affect those disulfide bonds in proinsulin. So the disulfide bonds are named like this, B7A7, which means the 7th amino acid on the B chain is connected with the 7th amino acid on the A chain. 7th amino acids in both cases will be cysteine, so a disulfide bond. Likewise, B19A20, and then there is an intra-chain disulfide bond. Today, we are going to talk about this one famously called Akita mutation, which is the CA7Y, where the cysteine is replaced by a tyrosine. What happens here? There is a mouse model called Akita model where we observe that it develops diabetes, insulin deficient diabetes. So what really happens here? Why does this Akita mice bearing this mutation develop diabetes? So we try to address this. So we collected the islets from the pancreas of the Akita. We extracted, we got the cell extract. We resolved them on the SDS page and did proinsulin immunoblotting. And we did that, we see proinsulin there. So the Akita, young Akita mouse is able to make proinsulin. So where is the problem? The problem is here. When we run a non-reducing gel, where we do not add a reducing agent, such as DTT or beta-ME, we see that proinsulin shows these oligomers. These oligomers are disulfide linked mostly. That's what we think, because when we add the reducing agent to it, it all collapses to make a single band. So these are disulfide linked oligomers of proinsulin. Dimer means two misfolded proinsulin come together. That's the dimer. Trimer is a three proinsulin structure, then a tetramer, pentamer, and so on. So this is disulfide linked complexes of proinsulin Akita that we observe here. So what is the significance of these disulfide linked aggregates? To answer that, we overexpressed wild type or Akita proinsulin in cells. What we observe here is very interesting. While the wild type is able to make proinsulin in the cells, and it is also secreting it, meaning proinsulin is going forward from the ER to Golgi. But in Akita's case, it only can make proinsulin in the cell, but it never secretes. We almost see zero secretion, which means proinsulin is staying back in the cell, and it is in the form of these disulfide linked aggregates that is settling back in the cell. It's not able to go forward. So this is what we learned from here. Akita mutant misfolds and forms disulfide linked aggregates. So now we wanted to address where in cells are they located? If they are staying back in the cell, where are they located? The answer is this. They stay back in the ER. We co-localize this with ER marker. For simplicity, I'm just showing the proinsulin staining. So the place where proinsulin is supposed to fold, they also misfold. Akita mutant misfolds, stays back in the ER. So that very well supports my cartoon. Proinsulin, that wild type, for example, folds well, moves forward, gets secreted. However, Akita misfolds, stays back in the ER, just like shown on that immunostaining, cannot go forward, cannot make insulin, and that could be a reason for the insulin deficient diabetes that we observe here. Now, that is not all. The mutant does more bad things. What does it do? I'll tell you here. So, human has only one insulin. Mouse have two. Mice have two insulin. Insulin 1 and insulin 2. Of the 4 insulin alleles that are available in Akita, there is only one of the allele on the INS2 gene that carries the mutation. Remaining alleles are totally fine. So the good alleles, they will make good proinsulin. The bad one will make misfolded proinsulin. So the misfolded proinsulin is somehow able to interact with the wild type proinsulin. We recently found that the misfolded proinsulin actually engages the wild type proinsulin using disulfide bonds again. Therefore, the picture I showed you in the previous slide, the ladder, which you can also see on the right top corner, that ladder is not just the mutant Akita, it's also a combination of mutant and wild type. So mutant and wild type aggregate together. And what happens due to that? The wild type is not able to move forward. So wild type export is affected. Therefore, the insulin making is affected, leading to diabetes in this condition. One other point to note, as I said, there are 3 alleles which are still normal. Only one is bad. So this is not a loss of function. This is a gain of toxic function that leads to this dominant negative effect of the mutant on the wild type. And that leads to less insulin making. So what we learned so far from the mutation story, when there is a MIDI mutation, that leads to misfolding, disulfide aggregation, trafficking problems, especially for the wild type proinsulin. So less insulin making, insulin deficient diabetes. So that's how mutation can lead to misfolding and beta cell failure in this form of diabetes. Okay, now let's go to the second part of the talk, which is related to the unfavorable folding environment. So what is it? Unfavorable folding environment. ER is the space where proinsulin folds, but it's not a standalone process. Proinsulin depends on several other proteins around it for proper folding. So what are those different proteins? Several of them have been studied, and we at this time when we were studying this, we wanted to really know which of these UPR sensors, unfolded protein response elements, or ER stress sensors, which of these proteins are participating in regulation of proinsulin folding. So we used a very simple approach. We added a drug to hit a protein, attack a protein, and then see if we see any effect on proinsulin folding. So we used a drug called PERK inhibitor. As the name suggests, we are going to inhibit PERK. So when it did that, we saw the same result what we saw with Akita, disulfide aggregates and ER retention of proinsulin. Next, we targeted this protein called BIP. BIP is a very well-known chaperone that facilitates protein folding. We killed BIP using this toxin called sub-AB. Within a matter of 2 hours, most of the BIP is gone. More than 70% of the BIP is gone. And at that point, majority of the proinsulin goes into this aggregates. While the BIP is still active, there is not a lot of aggregates. So this tells us if the ER homeostasis is perturbed, if the folding environment is not favorable, proinsulin will misfold and form these disulfide aggregates. All right. So one important thing we have to understand here, there is no mutation here. All the examples, the example about MIDI, the Akita that we saw before, that is a structural defect by a single point mutation leading to misfolding. But in this case, we are talking about wild type proinsulin. And wild type proinsulin by itself can get into these disulfide aggregates if the folding environment is not feasible, it's not favorable. These two examples I show you on this slide, they are pharmacological, they are artificial. We add drugs, we see some effect. Do they carry any physiological value? To answer that, we use type 2 diabetes model. Because in type 2 diabetes, the endoplasmic reticulum is morphologically affected and it's also functionally compromised. So that's a good place to look for proinsulin misfolding. So for that, we use the DBDB model, which has a mutation on leptin receptor. And so the leptin hormone that is supposed to inhibit hunger cannot bind to that receptor. So this type 2 model cannot detect hunger. So it keeps eating and eating. So blood glucose goes up and the beta cells start slowly failing because they can only make as much insulin. After some time, they'll start dying. That is exactly what you see here on in the picture on the right. There is immunostaining of pancreatic section from DBDB or control mouse. In the control, you see a lot of insulin positive cells. In the DBDB, there are very, very, very few insulin positive cells. So as I said in the intro, one of the introductory slides, insulin is the product. So precursor is proinsulin. When we see the product is so less, we thought the precursor will also be less. We may have less proinsulin in the DBs. But no, the answer was opposite. We saw more proinsulin actually in the DBDB compared to insulin. That was really fascinating. We were not sure what's happening here because there is more precursor made, but the product is less. One other feature, the localization of proinsulin. While in the control, it goes mostly to the Golgi, which means it has crossed the ER. But in the DBDB, most of the proinsulin is located in the ER. This is something we just saw with the PERK inhibitor. We also saw with Akita. And in those cases, we saw those disulfide aggregates. So we were wondering if type 2 diabetes also shows that. So to answer that, we collected islets again, but in this case, it's from the type 2 diabetes model at different stages of diabetes. So I'm showing you different glucoses, 100, 200, 500. So here is the immunoblotting of proinsulin from the cell extracts, from the islet extracts. What we see here, when we look at the very early stage in diabetes, 100 glucose, the mouse tries to make as much as proinsulin and insulin as the control would make. Okay. So why it's making? Because the blood glucose will slowly start rising. There is some problems happening with the beta cell. So initially it's trying to compensate for that by making more and more insulin. But there is a stage we have to note here, which is around 200 here. There is a lot of proinsulin, the precursor, but the product is missing. Insulin is down. And why is that? Same answer. Proinsulin gets into these disulfide link complexes at the expense of insulin making. Majority of the proinsulin is entangled in these aggregates. So it's unavailable to traffic forward, make insulin. That's what we think is happening. One other thing I want to highlight is these disulfide aggregates happen much earlier in the disease when the glucose is still normal. 101 is like a normal glucose range. At that stage, we already see these disulfide complexes, telling us that the misfolding and aggregation of proinsulin is preceding the onset of type 2 diabetes. So that way, proinsulin misfolding itself could be used as an indicator or a biomarker for type 2 diabetes. So that again supports my cartoon. Misfolded proinsulin stays back in the ER, we just saw, and so insulin making is affected. Okay, so this part of the work was published in eLife in 2019. This attracted like a lot of collaborations and many citations. If you're interested, please read this paper and get back to us with questions, ideas, suggestions. All right, so now I'll go to the last part of the talk. Now, so far what we learned, insulin gene mutations can lead to misfolding that can affect trafficking. Lead to insulin deficient diabetes, more like a type 1 in the Akita case. Okay, on the right side, ER folding environment problems like in type 2 diabetes. we have misfolding also. There again, we see trafficking problems. What I'm going to say you tell you towards the end is totally different. I'm saying that trafficking problems can lead to misfolding problems. So this was really surprising to us because trafficking is a step that happens after uh, folding step, but this is somehow affecting the folding process. This is something we observed very recently. We don't know how to explain this, but I'm just going to share a few evidences with you, and then we'll open the session for discussion. Okay, this is the first evidence. This is the collaboration with Miriam Knoop in Belgium and Timo Otonkonski in Finland. They are experts in making stem cell derived beta-like cells, islet-like clusters. And in this case particularly, they are looking at neonatal diabetes patient mutations on a certain gene called IPF5. IPF5 is a gene, it's a IPF5 protein. The function of it is to help cargo transport from the ER to Golgi. So it facilitates protein transport. If there is a mutation here, the ER to Golgi trafficking will be affected. So what do we see in these tissues? So we are comparing here wild type and the EPF5 mutant. We see something very similar what we observed in DBTB. There is more proinsulin and there is less insulin. Also, we observe that the proinsulin is retained majorly in the ER. It's co-localizing many of the cells with the ER marker calreticulin. So this ER localization is possibly due to the ER to Golgi trafficking defect. So Miriam Knoop was curious, we were curious to see what happens to proinsulin folding when this phenotype is observed. This is what we see. Then we run our regular immunoblotting for proinsulin from the extracts from this EPF5 or the control. We see these disulfide-linked aggregates of proinsulin once again. I have to point out that this new part of the study, which we recently developed, we made some technical changes in our immunoblotting approach. Now we are able to see misfolding at the monomeric level. So we wondered where this dimer comes from. Where does the misfolded dimer comes from? It comes from two misfolded monomers, right? So here we are able to show that there are at least two populations of proinsulin that we can detect. One is the native proinsulin that has the disulfide bonds intact and that makes insulin, while there is a non-native population of monomer that is shown with this red arrow. So with more buildup of non-native proinsulin, you will have less and less insulin. That's what we are observing here. And also the native proinsulin goes down in the IPF5. So just to summarize this part, IPF5 mutation will lead to ER2 Golgi trafficking defect that leads to a misfolding, secondary misfolding problem. For many years, actually for decades, there's a very commonly used drug called brifaldin A that blocks ER2 Golgi traffic. Many protein transport studies have been done using brifaldin A. So it basically, prevents the COP2 vesicle formation. So Golgi collapses on the ER. All of the cargo that's supposed to go forward are stuck in the ER. So this is much easier way to study the transport problem. So we did that. We used brifaldin A on the beta cells. We isolated mouse or obtained human islets from Prodo, treated them with brifaldin A. This experiment is particularly interesting because what we did here was we first treated the cells with cycloheximide CHX, that is a protein synthesis inhibitor. So we remove all the old molecules from the cells. Now, then we wash out the CHX and we are only concentrating on the new molecules. So at that point, when we finish at the end of washout, we add either the vehicle or the BFA, brifaldin A. When we do that, we observe that the brifaldin A leads to very tiny bit of non-native monomer and a lot of dimer. So those are the first species to appear as soon as the trafficking is blocked. So this tells us that the non-native monomer could be a key intermediate or the precursor to making that dimer. We observe the same thing with human as well as in the mouse. So this tells us that blocking the trafficking will affect folding. Now, one interesting thing about BFA is that it is reversible, meaning if you wash out the BFA, the trafficking will be fixed. So with that in mind, we ask this important question, very important question. If we restore trafficking, can we restore folding? Because we just learned that if we block trafficking, it will result in misfolding. So if you restore trafficking, can we fix folding? The answer is yes. We got a very beautiful data from this. So BFA addition leads to this dimerization, as I showed you, and there is also non-native monomer, and there is less of native monomer. Now, when I wash out BFA, because of its reversibility, thanks to that, the dimer is completely vanished. So you can see this on the graph plotted on the right. As we wash out, the dimer vanishes, non-native monomer goes down, and the native monomer suddenly comes up from nowhere. So we think that the conversion of the misfolded species back to the native disulfide bearing protein, that is something really new and important. So we understand from this that blocking trafficking will lead to problem. If you do not do a washout, you will just see the same result. Those are the third data on the graph. But if you wash out or remove the traffic block, you fix folding. We also learned that proinsulin misfolding is reversible for the first time, at least to my knowledge. Nobody has, although this is like a very controlled small experiment, in the long run, this would be an important result for in the field of diabetes. This part of the work was published in March 2024, where we highlighted that proinsulin folding, if there are problems with that, it can affect trafficking, and or if there are trafficking problems, that can affect folding. So these are tightly linked events. We don't know which problems comes first in diabetes. There is a feedback loop of defective ER homeostasis in the beta cells, and this is clearly implicated in diabetes. So what I showed you so far, insulin gene mutations can lead to misfolding. Unfavorable folding environment can lead to misfolding. I showed you two different diabetes models that support that idea. We see disulfide aggregation of proinsulin. We see diminished proinsulin and insulin in those cases. Both can lead to beta cell failure. I didn't talk much about ER stress. Okay. Now, finally, I'm introducing proinsulin trafficking problems. And I also showed you towards the end that if you fix trafficking, we can fix folding. But if these problems persist, gene mutations, folding environment problems, or trafficking problems, that will lead to misfolding and diabetes, or implicated in diabetes. So with that, I would stop and thank all the people in my lab. So that is the majestic view of the Michigan hospital I see from my home. As I step out of my apartment, I captured that picture. And then all of my lab mates, Peter, we sometimes wear similar T-shirts and just plan. And with Peter, it was not the case. We just happened to wear Michigan tees one day, and then that's a picture. And then there is Dennis, and there are many other trainees and friends in the lab. And this is the picture on the left bottom is a very recent one. Like a few days ago, we had a lunch party. So that was that. Yeah, so I'm thankful to all of them, and thank you again for inviting me, and to all my collaborators as well. Without them, we would not have been successful, and the funding agencies, NIH and fund. With that, I will stop, and I'm open for questions.
Thank you, Anoop. This is really interesting work, and we're really excited to have some questions from the audience. You can drop something in the chat, or you can raise your hand, mute yourself. Anything is fine. Waiting, anybody? Okay, I have some questions.
Sure.
I just wanted to, oh, someone has a question. Sharon. Go ahead, Sharon. Why don't you unmute yourself and ask?
Hi, Anoop. This is a great talk. I really enjoyed that.
Thank you.
So we've been talking about that, me and you, a while ago. So just to summarize, according to what you say, there could be a misfolding either due to mutation, like you saw in the Akita mouse, right?
Mhm.
Or ER stress or trafficking. But what about post-translation modification that can happen on cysteines? Now, you say the cysteines usually are not free, right? But this is in the proinsulin. What happens if in the proinsulin, which is the time where the cysteines are free, what happens if there can get any post-translational modification that will prevent this sulfur disulfide bridge formation? What do you think about that? Could that be?
So pre-proinsulin, when it starts translocating into the ER, there is like co-translational folding. Folding starts happening then after the signal peptide cleavage, and that's when the disulfide bonds form, and that's the proinsulin final. As with the proinsulin, they don't form intermolecular connections, intramolecular, I mean. So when there is like in the pre-pro form, they are mostly like open chain.
Right, and then they are very sensitive to any insult that could do any
They
PTM, right?
generally are degraded. Like if they stay outside the ER, they will immediately get proteasomally degraded. So they have to quickly enter the ER and fold to proinsulin. I think there is one or more mutations that prevent pre-proinsulin from entering into the ER. So those can result in accumulation of pre-proinsulin in the cells. But nothing more we know about the disulfide bond formations in a pre-pro at the pre-pro stage.
Thank you.
Thanks for the question.
Great question. Prasanna, Krishnan, do you want to unmute yourself?
Oh, yes. Thank you for inviting me, Anoop. This was a great talk. I was a couple questions. So just following up on the last question. So the non-native form of the proinsulin is a open form. Is that what you think? Like where the cysteines are exposed?
Yeah, actually there are 3 disulfide bonds, right? So even if one thiol is free, I will consider that misfolded.
Okay. So you could potentially demonstrate that like using like PEG maleimide or some sort of something
Yes.
attack
Yes, we already did that. In the eLife paper, we showed that AMS is a drug, sorry, not a drug, it's a small molecule that can bind to free thiols, and we see a shift there. So telling us that, so depending on how much you reduce, if I add a reducing agent and make it completely reduced, then you will have more AMS binding.
Thank you. And then 2 quick questions additional. One is, I wondered about the time course, like for your Berfeldin washout experiments, what was the time course from when you washed away the Berfeldin and did the Western blots? And then the second part was, how do you think that ER export might interact with the unfavorable protein folding environment in the ER. For example, I'm wondering if the two might actually be connected so that if you block ER export, you're actually creating an unfavorable environment. So ultimately it comes down to the unfavorable environment. So those are the 2 questions.
Yeah, the second part I will answer first because what you said is correct. Some trafficking defect happening here is actually creating ER homeostasis problems, and that's linked. So basically it's the unfavorable folding environment, but the first problem is trafficking here is what we think is, the first question was time for the Brifeldin. Okay, so Brifeldin treatment, as early as 10 or 15 minutes you treat, you already start seeing those aggregates, a dimer at least. But if you treat longer, up to like 1 hour or something, you see those complexes, and you wash it out immediately and leave it for half hour or 1 hour, it's fixed already.
Sorry, I pressed the wrong one. So what I was wondering is after you wash it off, oh, you mean it's fixed, like meaning already you see a reverse and you lose all those dimers and tetramers, etc.
Yeah.
Oh, wow, it's so quickly reversed.
Yeah, exactly. And so this dimer is the interesting thing. We don't know about the bigger complexes. When we go for trimer and more, it may be the dimer can go either ways. It could come and make more native monomers, or it could go the other way. It could go make the complexes. So the dimer is the thing that is doing the probably reversing is what we think here. Yeah.
Thank you.
Yeah. Thanks for the questions.
Next question. Great questions. Next one is Sam Stevens. Nice work, Anoop. Why does blocking ER Golgi trafficking result in misfolded proinsulin? I would have thought extra time would allow the PDIs to fix non-native disulfides.
Yeah, hi Sam. We talked about this over a poster a few years ago. This is a good question. I currently do not know what's happening. As Prasanna pointed out, we were just discussing. This could lead to a ER homeostasis problem here, right? So that is a thing. And as you said, if you leave extra time, yes, it may be possible. With Brifeldin A, as I mentioned, it happens within a matter of 10 minutes or 15 minutes. So I don't know what's the thing that's leading to this immediate effect. In Brifeldin A, I kind of understand that it's like some kind of ER stress or something due to the ER morphology problems. When the Golgi collapses on the ER, something's happening, but it's very, very quick. Yes, extra time, I'll have to think about how to design that experiment. But in this simple experiment we did towards the end, where we wash out the Briffeldin A, the thing seemed to quickly fix actually in the beta cells. But that's an interesting question, and at the moment, I really don't know the answer. If you block trafficking, why really the folding happens, misfolding happens? Yeah, but nice question. Thank you.
Anoop, I just dropped this paper in the chat, but you had a paper in 2014 elucidating the role of disulfide bond on amyloid formation and fiber reversibility on somatostatin 14, relevance to storage and secretion. That was, from the Indian Institute of Technology in Mumbai. And
Right.
I just wondered if you could comment on it appears that there's some kind of similarity here, right? In this, in the way the somatostatin is behaving and the way that the the way that insulin is also affected in this trafficking issue. Is that, can you compare, sort of contrast?
Yeah. This is an interesting question actually. Like no one has asked me before, but it's a good one. Okay, proinsulin disulfide bond making and its folding, misfolding, most of it happens in the ER. And that's what we are trying to talk about here and how that affects its trafficking and all that. The somatostatin that I worked during my PhD time, there is one disulfide bond in somatostatin. It's a small cyclic peptide. And what we found, those were all biophysics and structural biology studies. What we did there was we removed that disulfide bond. As soon as we remove the disulfide bond, somatostatin forms massive aggregates, amyloid-like structures. Something similar to what we see here in proinsulin. When there is a free thiol, there is this aggregation. In that case, it's amyloid-like aggregation. It's a different ball game. In proinsulin, I don't know. There are reports that say that it cannot form amyloid because the C peptide is like flexible and it prevents its amyloid fibrillation. In somatostatin's case, it readily forms amyloid, even with the disulfide bond intact. In fact, they form really beautiful amyloids when the disulfide is lost, and they don't form so beautiful amyloids when the disulfide is still there. They still make amyloids, but of a different shape. And so we were able to understand the conformational dynamics and structure and the importance of that disulfide bond in somatostatin in amyloid fibrillation. And those were really interesting studies. There was a 1987 paper where in the guinea pig brain, they observed open somatostatin. There is just one paper long back, but they say that it's real. We don't know for sure. That's just one study, but there is a possibility if there is an open structure of somatostatin, there could be amyloid fibrillation problem. Also, from the pharmaceutical industry point of view, somatostatin analogs are being made, and we need to have, just like insulin, we need to have something that is stable. There was also a time when they were trying to make open chain somatostatin as a drug, not very useful. It was easily degraded in the plasma. So so those are related to that study, which is mostly biophysics and structural biology. But now we are talking about biochemistry, cell biology of proinsulin. But there are some similarities. Thanks for bringing them together, Monica.
Yeah, that's very interesting because you kind of think about like the pathological misfolding of insulin and beta cells and the misfolding, that happens in neurons. The fact that neurons and beta cells have sort of that commonality in their neuronal precursor state. And Rita, just one more question. Rita Lahemsa and her group had a really interesting paper recently about microRNAs that are upregulated in type 1 diabetes and 5 that are upregulated, 1 is downregulated. And do you see, or have you seen sort of like in related literature, any impact of microRNAs, in this process?
Yeah, we happen to study this with one of our collaborators in Michigan Tech. We found, I don't blanking on the name of the microRNA. We found that that controls proinsulin folding somehow. I did observe the misfolding of proinsulin in that particular study. It's already published in scientific reports a few years ago. That was the only study where I worked with this microRNA thing. And I do think that it affects insulin production, at least in the beta cells. We didn't report much about proinsulin folding in that particular paper, though.
Okay, yeah, there was, I dropped one of the figures from one paper that kind of talks about the microRNA 133A involved in the whole processing of T2D. So who's anybody interested, they can take a peek at it. But yeah, this is really fascinating, Anoop. Thank you so much. I mean, the Arvan Lab is a powerhouse in Michigan, obviously, with a long history of inquiry into this realm. And it's fantastic that you're bringing out these new discoveries. We'll have to keep an eye on what's next coming from your group.
Sure. I thought Mike had a question. Should we?
Oh, yeah, sure. I'm sorry. I missed him. Yeah, Mike, please unmute.
Hey, Anoop, can you hear me?
Yes.
Yes.
Yeah, great presentation. I guess I just wanted to ask a question about like, how do you think we can reverse the misfolding that you're seeing, like that you see in T2D, so that doesn't occur due to any mutations? Like, what do you think is the limiting factor? Like, do we need to slow down translation, or do you think there's a way you could speed up the ER to Golgi trafficking to, since blocking it apparently is bad, could you speed it up somehow to restore insulin folding, like in the disease setting?
Both, actually. Increased translation, such as seen in T2D, can lead to like hypersynthesis induced folding errors can occur, and that would be a problem. And as you pointed out, facilitating trafficking is actually good, but we need to find out how to do that exactly. Like for example, say we add glucose, and what would happen here? It could also lead to increased translation on one hand, and that will lead to a problem. So we have to come with some ways how to facilitate the transport. There is other ways we have been thinking about. There are some enzymes, we didn't talk about this, oxidoreductases. These are enzymes that are in the ER that facilitate disulfide bond formations. If we are somehow able to up their activities, maybe we'll be able to promote. There is a paper from Jordan, it's not my paper, it's a paper in Arvan Lab, from Arvan Lab. Jordan Wright is the first author. There we find it was shown that overexpression of ER1 alpha, which is an important oxidoreductase, facilitates forward transport. So that way you are able to facilitate folding and also facilitate trafficking. You should understand one thing, that there is no benefit by transporting the mutant outside of the cell, right? Even if it goes out, it can make insulin, but the mutation will still be there, and it will not be biologically active molecule. So no, but what we want to selectively transport out is the wild type proinsulin. So it's kind of tricky when we talk about that.
Well,
but
it might be okay to dump even dumping the misfolded insulin could help alleviate the stress on the cell, even though the insulin's not active.
Yeah. So that way, improving the proinsulin degradation process is the thing. But once again, we want to specifically degrade the misfolded species, not the good ones. So yeah, but these are really good points. Thank you.
One last question here from Prasanna again. Is there a specific ER export receptor for insulin, such as SIRF4, that you can knock down to see if you get the same effect as you observe with BFA? And alternatively, will you try H89, which is another reversible chemical inhibitor of ER export?
H89 I have not tried. SIRF4, there is already a paper in Nature Communications, maybe. It interacts with proinsulin, so that's been published. But other molecules we tried, which I didn't discuss today, it's called monensin. So that blocks it little later. It doesn't block like BFA does. It blocks it later, so we can catch the native proinsulins there. So that is another measure of how to find native proinsulin in the cell. We have tried that drug, but nothing like Brifaldin A. Yeah.
Sounds like there might be some room for collaboration there.
Yeah, of course. Thank you.
Great. Okay, last call for questions. Great paper, great presentation. Thank you, Anoop. Have a great rest of your day, and we'll be in touch again soon.
Thank you. Bye.
Bye-bye now.